Thank you very much for that nice introduction. I hope I live up to that. That was quite a long introduction. Thank you, Dr. Edwards. So thank you, Dr. Asthana, Dr. Sterling, Dr. Lisa, Dr. Kine, and also Dr. Golden for this wonderful invitation here today. And let's see if I can operate the clicker. Okay, great. So brain health, looking back to move forward. And I have my Twitter up there if there's anyone doing live tweeting. I don't know if folks know about live tweeting. That's a new thing now when you live tweet little research updates. So I just learned about that this past summer. Okay, so I'm starting off with a photo that's actually kind of fuzzy. And why do I have this photograph up here? Well, this is a photograph of my parents who actually both went here. They both went to Madison. And they actually got married here in Madison. I was born here. Their wedding reception was at Union South. And so I have deep roots here, and I have not been here since we moved away when I was age 6. So it's been quite some time. So it's really very special and very unique to be back here. And we had a funny conversation last night about the Blue Moon flavor ice cream. They thought I had made up that memory, but I went and I confirmed it at the counter at Babcock's in Union South, that Blue Moon is a real thing, though I had actually recalled it as Blue Moon chip. So I don't know if they had chocolate chips back then when I was age 4, 5, and 6, but it is seared in my memory. So I definitely have deep roots here, and my father is actually from Beaver Dam. And another really interesting historical twist is that my father was actually in the building during the Sterling Hall bombing. He was working in a physics lab in the middle of the night. So we actually have some connections here to some major historical events. So it's great to be back here. And don't worry, I'm not scared of the snow, even though I have been in California for quite some time. I did grow up in Western Massachusetts. I can deal with snow, so everyone's quite worried about me. Okay, so today I'm going to be talking really about life course epidemiology and how some of the work that has emanated from my group and others has really led to this reconceptualization of dementia as a life course process. I will also be focusing on vascular risk factors, and for each of my themes, I'm going to touch upon one of the 4 cohort studies that we've been doing. So these are in-the-field studies that have been informed by the epidemiologic population work that we have been doing, leveraging the Kaiser Northern California data. I will also be talking about brain aging specifically for type 1 and type 2 diabetes. And also a huge focus of my work is really understanding disparities, so trends across different racial ethnic groups and what we can do to change that. Hopefully you'll agree with at least one of those. So, it's really complicated right now in the field of dementia and Alzheimer's disease. We are arguing about the very definition of what Alzheimer's disease is, and we're really in this historical time point. They're about to come out with new diagnostic criteria for what defines Alzheimer's disease. So it's sort of challenging, but at the same time, it's incredibly exciting. So just in the last 10 years, and a lot of it has been fueled by the brilliant work coming out of here, we've been able to image the brain in vivo in ways we've never been able to do before. We can see amyloid now when people are alive. We can see tau. We don't have to rely on people passing away and their families agreeing to donate brains and looking at brains postmortem. So it's a really, really exciting time. So it's challenging, but it is really great. So I'm not going to spend too much time on this. You could dedicate a whole day to this. And I know I'm preaching to the choir, but we're still grappling with our definition. So dementia does not mean a disease, it means a syndrome. But the 2 most common subtypes are Alzheimer's disease and vascular dementia. And when I started in this field, there was a definite line. People really felt there was Alzheimer's disease and there was vascular dementia. We now know that those lines are actually very blurry, and the majority of folks who are diagnosed with probable Alzheimer's disease, if you do get the opportunity to look at their brains, they have evidence of mixed dementia. So this is a condition that is very heterogeneous. And again, there's been a lot of great work from here that's really well characterized that heterogeneity of what it is. But I think we're also at the tip of the iceberg. I think in the next 5 to 10 years, as we dig deeper and deeper and really see, what do brains look like that are behind these people getting diagnosed with dementia, we're going to find out more and more. And of course, as was touched upon by Dean Golden, it is highly, highly important. If things do not change, we will have 115 million people with dementia worldwide. That is huge. That is enormous. That sign that down there is a sign of a tsunami warning. You can see those on the beaches in California. We call this the gray tsunami. It is a tsunami coming of dementia cases. So there's never been really a more critical time to dig in and really take this head on. So why do I care about the life course? Well, as we know, these changes in the brain can happen over many decades. And when you only study people very late in life, you get worried about this thing called reverse causality. It's hard to know which came first. So, for example, if you're looking at folks who tend to have really high cholesterol and they seem to have worse cognitive function, it's really hard to know sort of which came first. If you only study very elderly people, you also worry about selective survival. So, of course, if you are getting diagnosed with dementia and you are elderly, you have survived. You have survived to an age where you can get diagnosed. And that is really critical for taking a step back and understanding how competing risk of death and competing risk of mortality from other diseases and other risk factors, and how those risks of death and mortality are different across different subgroups of our population. So you can get very different answers depending on who you study. And when you can get very different answers, understanding risk factors, looking at different groups of folks. So for example, men, for example, are not as likely to live as long as women and much more likely to die of a vascular event. So what does that mean when you're looking at the association between vascular risk factors and dementia in a group of elderly men and women? Okay, it means something different. It means those men with those vascular risk factors are very unique. They are more of a selective survival subgroup, for example, than the women. So really appreciating how these things kind of work in concert with other diseases and other risk factors is really critical and key to understanding brain health. So we like to leverage electronic health data as a platform for informing our cohort studies. I'm using the word smaller studies, but these are still cohorts of about 1,000 or more. And we've done a lot of work looking at Kaiser Permanente out of Northern California. So just to give a really quick update on what that is. So an integrated healthcare delivery system means you receive all of your care in the same system. So you get a prescription, you go to the hospital, you get a diagnosis. It is all captured in one system. So it's a really nice living lab. So I just mentioned that. It actually was the first federally funded study, though officially it was like a little add-on to your well checkup. So if you were a Kaiser member in Richmond or Oakland in the 60s or 70s, there's a 99% chance that you were in the multiphasic. And you often had several waves of this data. So it was offered at all of your well visits during a certain time period. So there was a clinical exam, a fasting blood sample, and we did a number of cohort studies based on that. I didn't want to bore you too much with this, but we use Cox proportional hazards using age as the time scale rather than study year. That's a very robust way to adjust for age. It came out of the cancer epidemiology literature when age is very, very highly associated with both your exposure and your outcome. So when we're following people, we start to follow them January 1st, 1994. The way that the model works is this age, January 1st, 1994, and then age at the date you get dementia or you get censored or you die. And then we took into account a number of midlife and late life comorbid conditions. So using time-varying covariates to take into account not only if folks had these other conditions, but how long they actually had them. So our first work from this body of work was looking at high body mass index. Today it seems like, big yawn. We've seen this finding so many times that high body mass index in midlife is associated with dementia. But at the time that this was published, this was the first paper looking at that. And there was a lot of pushback at neurology conferences because one knows that when you're working with patients with dementia or Alzheimer's disease, they can lose weight, they can be very thin, they can be frail. And we had to explain that we were looking over the life course. So we were looking in midlife. And so even after taking into account all these other comorbid conditions, we see an association between being overweight or obese in midlife and dementia. Of course, the big talk at the time was metabolic syndrome, and we didn't quite have the right data to delineate metabolic syndrome, but we did have this great anthropometric measurement of central adiposity, so sagittal abdominal diameter, and it's used with a slide ruler, and it really sort of captures the distance. If you were to take a laser and go through your belly button and out your back, it's that distance. And lots of studies have shown it's actually a better marker of visceral adipose tissue, the really dangerous adipose tissue wrapped around your organs, than waist circumference. So the way this slide works is it's actually SAD to have a high SAD. Okay, so SAD is sagittal abdominal diameter. So in a nutshell, what this slide is showing you is that, of course, it's not good to be obese or overweight, but those individuals who were obese or overweight had about the same risk, about a 80% risk, as those who were normal weight but had a high sagittal abdominal diameter. So that central adiposity is not good. Now, of course, if you're both overweight and obese and also are carrying that weight around your midsection, then we have what's called a multiplicative increase here in risk factors for dementia. It was quite interesting. We got a lot of feedback when this work was published because a lot of people really felt like, if they were overweight or not taking care of themselves, so what, that might affect their heart, but knowing that it could affect their brain and their brain function in late life really spoke to people. And we still have folks, getting in touch with us today about that work. It has since been replicated in many different cohorts around the world. This is showing heavy smoking, and again, this seems really quite obvious, but actually there was a bunch of case control and cross-sectional studies that had suggested that smoking was good for Alzheimer's disease. You can go back and find these studies. Some of them may have been funded by the tobacco industry, but there was work at the time in the Parkinson's space really showing that nicotine might have a beneficial effect on the dopamine receptors, so people really believed it. But again, no one had looked long term, and you can imagine if you take a group of 85-year-olds with Alzheimer's and a group of 85-year-olds without Alzheimer's, it probably does appear as if smoking is protective, because if you're 85 and you're a heavy smoker, you are very magical. You have not succumbed to many things. You did not get a stroke. You are alive. So that is not the best way to ask these questions. We need to follow people over time. I'm not sure if folks have heard about the CADE study out of Finland, but they had the very first dementia risk score, which was based on midlife vascular risk factors. It was very exciting for the field, but everyone said, oh, but it's just, Finns. How representative, is that? It's a group of Finnish people living in a wonderful country with great healthcare access, great education. But we actually validated and replicated that score in our Kaiser Permanente cohort. And what was really beautiful was not only did it predict equally well, but it really predicted quite well for our non-whites. And so that was really an important thing for the field to understand, that really these mostly modifiable risk factors for midlife are predictive of dementia 30 to 40 years later, and not just for Finns. Okay, so midlife matters, but what else? Does it matter going back further? Does young adulthood matter? And how do you take into account the bias of simply surviving? So different groups of folks tend to live and have different life expectancies. So how do you take that into account when you're understanding these associations between risk factors and disease? Are there critical windows? So is there a certain point in the life course, where hypertension might elevate your risk more than someone else? And so really what it comes down to is how you frame your cohort really can affect how you are able to ask your question. So coming back to what is the role of risk factors before midlife, some work led by one of my mentees, Dr. Paula Gil-Sans, looking at very early onset hypertension. So this was looking in early adulthood and also mid-adulthood. So mid-adulthood was from 40 to 50, and early adulthood was from 30 to 40. And there really had been a dearth of work looking at this, particularly by sex, so separately by women and by men. And so the story here is that by midlife, being hypertensive significantly elevates your risk if you're a woman. Pre-hypertension does not seem to have an association. And earlier, in early adulthood, there also is a signal in women for being hypertensive and elevated risk of dementia. And you can see here we don't see the same thing in men. And one of the big things is that we're only able to look at men and women who had hypertension, but they survived. They survived to 1994 when we could start following them for dementia. So what does that mean for these results? It could mean that maybe hypertension early in life is not really a risk factor for men, or maybe if you're a man and you had hypertension early in life and you survived to late life, it does not elevate your risk. There is something very unique about you. And this is one of those examples where we really dug into innovative methods to try to account for this survival bias. So we actually did inverse probability weighting, where we created a pseudo population and we calculated weights for those who had survived. So they started off in Kaiser, they survived to 1964, they could be in the MHC, then could they survive to 1994? And then, of course, during our follow-up in the model, we're taking into account survival. So we really tried hard to scrape away at this question about selective survival the best that we could. And this is just showing you that if you're a woman with high blood pressure in early adulthood, this is what your cumulative incidence of dementia looks like up until age 60. So you've survived till 60, and then we're looking at incidence of dementia. So it seems to suggest that midlife matters and also early adulthood matters, but these questions about sex differences in the competing risk of death and mortality and succumbing from other things during follow-up, I think is a very, very tricky question that we really need to take a very innovative approach to. So we have a nice opportunity to look at vascular risk factors over the life course. We have several waves of data starting in 1964, and Dr. Elizabeth Rose Maeda, who's assistant professor at UCLA, has now written a grant to really dig into what are the long-term patterns of these vascular risk factors for dementia and for cognitive aging, and really digging into some of the racial ethnic differences here and taking into account that selective survival. Okay, so I want to talk about one of our cohort studies, because this work really excited us when we were using this electronic data to leverage and ask questions. It got us very excited about doing a cohort study. So the CANDLE study is funded by the National Institute on Aging. It is 1,700 people, and our goals were to look at ethno-racial differences in dementia incidence and rate of cognitive decline, to leverage these 50 years of prospectively collected data, which is very unique. But as I found out a few minutes ago with Sanjay's new grant, he has, it sounds like, 60 to 70 years of data going back. So that's quite exciting. To really ask questions about life experience, life course health, and early life. And are we going to see the same patterns by racial ethnic differences in dementia as we do in a cohort study where we routinely follow and evaluate people and see them on a regular basis? We also want to characterize brain imaging markers, so we are doing amyloid PET and structural MRI on 400. We really talk about what does diversity mean. When we're thinking about racial ethnic differences in this study, we're not really thinking about biology. We are asking a lot of questions to get at differences across the life course. And diversity doesn't only mean racial ethnic differences, it can also mean things like education. We have worked very hard to oversample those with very low levels of education. Folks with a high school or less level of education, again, tend to be left out of these research studies. They're harder to find, harder to recruit, and harder to follow. But it's important that they are also in these studies. So what are we looking at? We're using the SANUS, which was developed by Dr. Dan Mungus at UC Davis. It works beautifully in Spanish and English. He hasn't gotten a chance yet to validate it in Mandarin or Cantonese, which I feel sad about because my Asians tend to be higher educated in these studies because we have a overlap between level of education and fluency in English. We're also using the NIH toolbox. And we're developing an algorithm to see how we can use those scales as a trigger for future dementia incidents. We're doing the neuropsych test battery. And we're doing a huge number of psychosocial measures. So residential history, every home address, every school attended, perceived discrimination, perceived social status in the community and currently, stress and coping, and social support, as well as physical function. So this is our handout. You can help change the future of health. We had a lot of discussion of who would be on this handout. We want someone that really represents the diversity of the group that we are trying to recruit. And we also use infographics. So, often you get a letter explaining a study and it can feel very, very daunting. And the IRB requires you to put a lot of language in these letters, and so much language that someone with, perhaps lower levels of literacy or education can be completely turned off by being in one of these studies. So we try to use these study insets, which gives a really nice pictorial sort of overview of what happens when you're in the study. You're going to fill out a consent form. You're going to answer questions. We're going to play some memory games. We're going to measure some stats on you, and you will, of course, get paid. So we work really, really hard on that. So who is this Kaiser cohort? And we are currently in wave 2. So they are long-term Kaiser members. They were also part of the multiphasic, so we do have data from earlier in life. We've tried to oversample those with lower levels of education. We see them every 16 months. We are also doing a blood draw and extracting DNA and storing it. Haven't had time to think yet about leveraging that. We've been so busy being in the field. We don't have money yet for post-mortem brain donation, but we are actively seeking additional funds. So we hope. So that is our candle study. We're currently on wave 2, and if you're going to the AAIC meeting this summer, we did submit a whole host of abstracts on this wave 1 new data, so you'll see some more updates there. Another big part of our lab is digging into diabetes. And so this is a really large public health problem. A lot of the attention is focused on type 2, which is most of diabetes. But what's not as well known is actually type 1 is also going up in incidence. And it is an autoimmune disease that's highly genetic, and it's driven by a completely different set of risk factors than for type 2. So with type 2, it's pretty well documented that they are at higher risk of dementia. Whether they have more Alzheimer's pathology, I think, is a bit undetermined at this point. And we've sort of asked these questions: does appropriate glycemic control lower risk of dementia? Because we know that it lowers risk of other complications of diabetes. So we've seen associations between very high levels of glycosylated hemoglobin and low levels and increased risk of dementia. We were surprised to see this group here. They weren't very big, but who were those people that had exceedingly low glycosylated hemoglobin? So this is a marker of the past 3 months of effective blood sugar control. So who were those people who were very, very tightly controlled, maybe over-controlled, and why are they at higher risk of dementia? So that led us to our follow-up study to go back and pull from the records very acute low blood sugar episodes. So these are blood sugar episodes where you ended up in the emergency room or the hospital. This is not something that you would mention at a visit. So this is something captured in our health system. And we saw a very robust association between these episodes and future risk of dementia. And we even did some sensitivity analyses where we were looking at hypoglycemic episodes that had actually occurred 20 years or more prior to when we started following these folks for dementia. We know from the animal field that if you induce these very strong low blood sugar episodes, you actually blow out, part of the CA1 and CA4 part of the hippocampus, the seat of learning and memory. Of course, no one's going to do that to a human being in an experiment. So this is a question that can only really be asked by looking at observational data over the long term. So a very strong association. We were really lucky to host a Fulbright fellow, Dr. Lisa Exalto from the Netherlands, and she was really interested in a diabetes-specific dementia risk score. So we had these dementia risk scores that were for the general population, but can we look at diabetes-specific variables and come up with something where you can tell someone who has type 2 diabetes what is your 10-year risk of dementia? And so we worked hard on this, and she came up with this really simple point scoring system, which is something that can actually be done in regular medical care. So this could happen at a primary care visit through self-report. It can very easily be calculated with electronic health records, and it tells you what is your 10-year risk. And it's a combination of age, education, these very strong low or high blood sugar events, and it actually can tell you. So if you were to come in and see a doctor and say you had diabetic foot and depression and cardiovascular disease, and you were between 65 and 69, that gives you a score of 8, and you would say, okay, your 10-year risk of dementia is 50%. So we think this is a really nice tool. So I talked before about how type 1 is actually going up in incidence, and no one really knows why. If it's emblematic of the trend that we see overall in autoimmune diseases and what is actually going on. But they've been sort of, ignored in terms of aging and health research, because type 1s historically just weren't living as long. And now they actually are. So we've had these great advances with glucose monitors and the insulin pump. And so now they have a life expectancy that is almost the same. And so that is really great news. But the uncharted territory is, what does this mean for their brain aging? And what does this mean for their cognitive health? And so we actually have done some cognitively challenging tasks. So we wanted to look at longitudinal glycemic control in this population in our type 1s that are part of our Kaiser Northern California system. And this work was led by Dr. Beth Lacey, who is a postdoc fellow at UCSF. And what we basically found is, looking over a 15 to 20 year period, if you spent 70% of your time at over 8% of HbA1c, you had a significantly increased risk of dementia. This was just published earlier this year, I think in January, in Diabetes Care. So it's showing that sort of long-term exposure to these higher levels is not good for your dementia risk. So glycemic control matters. Work by Dr. Gilsans has looked at depression in this group. And so that is also a risk factor in and of itself for having a very, very strong low or high blood sugar episode. And so these are really complicated patients to treat. So you can imagine, if you're a geriatrician or a primary care doctor or an endocrinologist and you have an elderly patient with type 1 or type 2, there's a lot of stuff going on. And they are, as a population, at higher risk for some mental health problems. And so I think it's really important, as a field, we appreciate the complexity of this population and really try to understand how can we make their cognitive health the best for them, and how can they think about successful aging. There seems to be this window of time shortly after you are diagnosed with depression, and again, this is among people with diabetes, that they seem to be at the highest risk for one of these very, very strong low or high blood sugar events. So it's hard to know, are they responding to treatment? Is it feeling that they're depressed and they can't manage their diabetes? So we're trying to dig in a bit more and understand this. Okay, so we know that there's this bi-directional association. And so if you have depression, you're more likely to have one of these very severe events, and also vice versa. So we're going to skip ahead here. So we have our study of longevity in diabetes. We're following 1,200 people, 800 with type 1, 200 with type 2, and 200 without diabetes. And we are doing a very in-depth cognitive neuropsych battery on them to try to understand, what does cognitive aging look like in this population, cognitive function. Okay, we have 10 more minutes, so I want to talk about ethnic and racial differences. So I touched on this a bit before. We published a study in 2016 led by Dr. Elizabeth Rose. And what had been known prior to that study was that African Americans are at higher risk. Caribbean Hispanics are also at higher risk. We knew about Japanese Americans, and it looked like the risk was about the same for Mexican Americans. No one had really comprehensively looked at all different racial and ethnic groups. So we decided to look in 274,000 Kaiser members over almost a 14-year follow-up at incidence of all-cause dementia. So we did a number of different adjustments. We did our ages time scale, we looked at medical utilization, and then we looked at a whole number of comorbid conditions as well. So 20% were diagnosed, 43% died during follow-up, and our average follow-up time was about 8.7 years. Okay, so this is just showing you our age standardized dementia incidence rate. So we see here the same story, that African Americans have a higher risk, though the difference between African Americans and whites isn't as large as in studies coming from memory clinics. Our Asian Americans had the lowest rate and risk. And this is the first time that this group had been looked at comprehensively. There have been beautiful work coming out of the Kame study and the Haas study, but those studies are both exclusively Japanese Americans. And in Northern California, we have Asian individuals from many, many different countries historically. This was also the first time that American Indians and Alaska Natives had also been looked at, and they also show a higher risk. Okay, so in comparison, we can see here that our African Americans have a 70% higher risk, our Native Americans 42%. The reference group here are Asian Americans. So why do we care? Well, one of the things that we took home from this was the size of the differences between these groups was actually smaller than had been seen in some of these studies that were not population sourced, but were sourced from memory clinics. It was the first time anyone had looked at Native Americans and Asian Americans comprehensively. And just to give you an idea of what it would look like if everyone had the same rate as the Asian Americans, you would actually have 190,000 fewer cases of dementia annually if on a population level everyone had the same rate as those Asian Americans. So of course the next question is why? And Asian Americans are a very heterogeneous group, and Dr. Elizabeth Rose has some follow-up funding where she's actually going to be digging in to these subgroups and looking at genes and really trying to ask this question, because it would certainly help everyone if we had a lower rate. So one of the things that we don't know about is why are these changes there? Why is it that some of these groups have a higher incidence of dementia, but their cognitive decline is not actually steeper? And what is the role of early life risk factors? So we think about race as a sociocultural context. And I'm going to skip ahead here. We've done some nice work looking at place of birth. So again, this is in our Kaiser group. So if you were just born in a high stroke mortality state, you had a higher risk, whether you were white or black. And these are folks who all were in Northern California by the 1960s and the 1970s. So what is it about being born in this state that increases your risk of dementia? Is it different educational value? Is it different cognitive reserve? So we're really trying to get at sort of these life course questions about health over the life course. We just published a study last week that was led by Dr. Gil Sans in Alzheimer's and dementia, where we used our electronic data and looked at incidence of dementia specifically at ages 90 and up. And you can see here our person years. So there were 771. And you can see here what was the adjusted incidence rate. And you see a bit of the same story that you see at younger ages. Our African Americans have a higher rate. Our Asians have the lowest. The differences aren't quite as large when you're looking at a 65 plus population, but the story looks to be the same. These are our Cox proportional hazard models. So you can see here the same story holds even when we take into account these midlife and late life health comorbid conditions. But the risk increase here is smaller than what we see at younger ages. So the differences are there, but they are smaller. And this is just showing you over time our cumulative incidence up until age 90. So if you survived until age 90, what does it look like? This is the things. I hope you believe in one of these by now. I hope I have convinced you. I want to thank all my collaborators and all my NIH funding. And I want to thank everyone again for the chance to speak here. I'm going to end with this.
About 10 minutes for questions, and we have people with mics. So if you have a question, please put your hand up and we'll get a microphone to you.
Yeah, I was just curious. The survey or the study that you did at Kaiser Permanente began as early as 1964. At that time, if I'm correct, very little was known about Alzheimer's. I'm not even sure if the term was used. It was just called senility or dementia. So could you discuss how that has evolved over the uh, longevity of the study? Thank you.
Yes, thank you for that question. Right, so from the 1960s and 70s, those additional clinical data and that additional, surveys and questionnaires that were given out, no one was thinking about old age or dementia or Alzheimer's disease. They were just saying, wow, here's a chance to get more data on folks and really sort of answer questions about screening within a integrated healthcare delivery system. The data has turned out to be a gold mine, and there's tons of studies looking at risks of lung cancer, breast cancer, cardiovascular disease, and now as the folks have gotten older, also looking at dementia. So that's a great point. Yeah. And we actually have a really detailed questionnaire about smoking, and that was behind the study that we did looking at pack years, over the life course, because we had these several time points of questionnaire data. Yeah.
Hi, that was a wonderful hour of talk there. So I'm just curious, in terms of recruiting essentially non-white people, it looks like you've had a considerable amount of success with that. Any thoughts on why you've been so successful in that
That's
area?
a great question. Yeah, so we've done a number of different things. One is our research staff are from the community that we're trying to recruit. It's really important to build trust. And so we had a lot of focus groups in the beginning of what would make sense and what would work. And yeah, we are thinking about different racial ethnic groups, but also different levels of education as well. Because as I said before, it's highly educated white urban dwelling rich white people is what has made up most of the big population-based studies. So we're trying to think about folks that live in more rural areas too, lower levels of education. But I think gaining trust and building trust, and we have had interesting things happen where there's been members of my team who may have a name, like their first name is very typical of someone from that community. And they will tend to do better on the initial recruitment and enrollment calls than someone else who's also from that community, but might not have a name that's as, used as frequently. So it can even come down to that. Um, I try really hard to build trust with them as well and be accessible as a PI, as an investigator on this. I can't always speak as being someone from these communities. I'm, obviously I am white. And so I can't pretend to be, from these communities, but I really think it's important to build trust and have staff from those communities. I think we had a question up here in the
Thanks, Rachel. That was a fantastic talk, as usual. I'm a big fan. First time caller, though. So I was wondering if you had any data on siblings, if you knew like family relationships, if there were any opportunity to look at that.
Yeah, I feel like, did we talk about this at the AIC? I feel like we might have, or family members or spouses. So we do ask questions about siblings. We do know family structure. We haven't leveraged, that yet. It's been so much work to sort of get these studies off the ground. There are opportunities with the Kaiser electronic data to look at family structure, and you can figure out siblings and also spouses. Unfortunately, it's not something that we have done yet. We're talking about it in some of the cohort studies about adding on, spouses or caregivers or other important family members that are part of the person's life. But yeah, that's a great point.
So you've identified episodes of severe hypoglycemia as a risk factor based on, I presume, an association. Has that controlled for education level or access to adequate health care? That may be other factors mediating the influence of such episodes?
Yeah, that's a great point. So we are doing that right now in the SOLID study. We're looking at associations between these really severe low blood sugar episodes and cognitive function. We're absolutely taking into account education and access. In that JAMA paper, the one where we were looking at acute low blood sugar episodes and risk of dementia up to 20 years in type 2s, we controlled for a number of things and did a number of, also stratified analyses as well. We did take into account medical utilization the year that they were diagnosed with dementia. We also we pulled prescription data, like not if they were just on insulin, but years on insulin. And then we looked at long-term HbA1c. So yeah, we really sort of tortured the data to see, but the association was still there. I think about it a lot because with the type 1s, it's a tricky thing because part of why they survived was because they over-controlled themselves, and they are more likely to have a history of these low blood sugar episodes. And so I would love to do some brain pathology and brain imaging studies because I don't necessarily think, it's a risk factor for dementia, but there must be, some kind of sort of learned response, from the brain because these people have survived even though they have had these episodes.
Hi, thank you for the great talk there. I had a question about, you had on that slide, innovative methods are needed. I was curious if you had any comments or ideas about capturing exposures around innovative methods and anything around, critical thresholds and exploring, kind of categorizing that exposure.
Yeah, so that's a big focus of our work now, led by Dr. Elizabeth Rose. She got a K99, which is now an R00. And so she is looking at sort of the 20-year, picture of these risk factors and looking at not only patterns, but certain inflection points. And she's doing it, separately for men and women to try to disentangle these sex differences that we see in both the dementia and risk factors, but also by racial ethnic differences as well. Because I think with some of the studies that have come out, I think we've gotten, maybe some of the wrong answers because some of these groups can have, really accelerated aging and they're much more likely to die at a younger age. So within those groups, who survived with those risk factors, I think is not emblematic of the bigger population. So she's really digging in and, we're doing both causal modeling, some inverse probability weighting, and we're also playing around with different ways to take into account the competing risk of death. Because there's very different ways you can, model for that. And so, what does that mean? It's really fascinating, and I'm glad I have a lot of folks in my group that are interested in it. And if you're interested in it, there's a group called Melodem, which is Methods in Longitudinal Dementia Epi, led by Maria Glimor, who's a good friend and a colleague of mine at UCSF, and Carole Dufoy. I think I'm saying her last name right, but she's in France. And they have these great meetings, and they're usually right before the AAIC or right after. They've always been in France, but this year it's going to be in Northern California because the AAIC is in Los Angeles. So, and you can get on, there's weekly conference calls, there's work groups, folks present works in progress. There's a really fun Melodem Twitter feed that's really fun. Folks share code and give tweets about some of the ways they are tackling these methodological questions because I think we're just at the start of really understanding it, and it's quite tricky, but very exciting, of course, but I'm completely biased.
Yeah, going back to the Kaiser Permanente study, you specifically excluded issues arising from AIDS. Was the reason for that because AIDS was largely or completely unknown until later in the study?
Oh no, so the way we categorized, and thanks, that's a great point. The way we categorized dementia was we took out HIV-associated dementia and alcohol-associated dementia because we wanted to, possibly look at those separately. We were really trying to get at sort of the mixed dementia, Alzheimer's dementia, vascular dementia. So those have a very different, etiological underpinning. So we didn't want to conflate them and look at them. I do have a colleague, though, at Kaiser who's doing some really great work. He just started very recently looking specifically at HIV-associated dementia and HIV-associated cognitive decline. I think it's a really very, very interesting area.
Thank you so much for your talk. That was a great array of studies. So my question is about the depression and hyperglycemic incidences. And I was just wondering if you could speak to what you think the biological mechanism connecting the two might be. And then a second side question, are you going to look at depression in your studies that are studying ethnic diversity? I think the one I'm remembering off the top of my head is your candle study, and potentially maybe coping mechanisms in different ethnic racial groups, or other thoughts on that. Thank you.
Yeah, great questions. So yeah, for all of the cohort studies, we are collecting symptoms of depression. We're not doing a formal depression, interview. Of course, if someone triggers and they seem to be very depressed or have suicidal ideation, we get in touch with their primary care provider. But we are collecting symptoms of depression. We will know who has diagnoses from the electronic health record. We have not dug into coping mechanisms specific to depression, but we are collecting some general personality measures. Like specifically in the diabetes study, we have sort of this locus of control measure that's very specific to diabetes, like my sugars will be what they will, or I don't think it really is important to check my blood sugar, or I don't have any say over my blood sugar, so sort of getting at, and most of these folks, not surprisingly, have a very high locus of control. They very much feel that they are in control. That's why they have lived to old age with type 1 diabetes. In the other cohort studies, where certainly there are some folks in there that happen to have diabetes, but it's not focused on diabetes, they're studies of, sort of a general elderly or aging population, we do have symptoms of depression. We are not collecting questions about coping with it, but I think that is something very important to think about as these cohorts age. And among those who do have comorbid disease, I think there are big sort of sociocultural differences in how one copes with the disease, how one takes care of a disease, and how one talks about symptoms. And I think your first question was about the work led by Pablo Gil-Sanz on depression among those with type 1 diabetes, and then subsequent future risk of a very low or high blood sugar episode. So yeah, what we see is there is this very strong association. It's the strongest within the 6 months after being diagnosed. So we've had a lot of discussion. Are they on a new medication that's interfering with their blood sugar control? Or are we sort of capturing a window of their life where they're very overwhelmed in those 6 months after diagnosis? Is the depression being caught sort of later stage, and these folks are just not doing well and not able to take care of themselves? So I think that's a really important question, because I think, as I mentioned earlier, these are very complicated patients, and we need to really think about the best way that they can have, also the best brain health. I didn't get a chance to go into it too deeply, but Dr. Gil-Sanz showed in her work that the reverse association also exists. So, having depression puts you at higher risk for these very acute hypo and hyperglycemic episodes. But people who didn't have depression, who had a history of hypo and hyperglycemic episodes, were also at future risk of depression. So we have things going on, at both ends. But I think in terms of clinical epidemiology and, sort of healthcare delivery of these patients, we need to sort of dig in and, find out why.